Good morning, everyone. Day 3 of the Sacramento River Drainage Spring Run Chinook Salmon Workshop on the state of the science for population estimates. This is Julie Lineback with Kearns and West. I'll be your facilitator for this plenary session today. For those of you who were not here on day 1 with us in the plenary, you are going to be muted upon entry, and this workshop will be quite large. We had almost 200 people on day 1, so the muting is to reduce background noise. So thanks for your patience with that. We do have discussions today, and we will be participating in the discussion via, I'll be calling on you, you'll be able to unmute yourself at that time. And in order to indicate that you want to join the queue, please use the chat function. So we will not be using the raise hand function today, just the chat, because some people don't have access to it. So I want to make sure that we're using the chat. So welcome, everybody. I know that in the past few days, it seems like it is a small victory just to be able to log on and join a webinar like this with the power shutoffs and everything that is going on. So thank you very much for joining us. I see that we have over 100 people joining this morning. So thanks for coming back after day 2, after you had all those rich breakout discussions. I want to say, now that we have a large number of people with us, that if you need technical assistance, I'd like to invite you to contact the technical assistance team. There's a whole team supporting this. The email is engage@deltacouncil.ca.gov. And the number is 916-798-9817. I want to also let people know that this day 3 is being recorded like we did for day 1, and that there is a workshop cap on the number of people that can be here together because of the limitations for the webinar platform, which is 300 people. So that is already fairly large. I see we're just over 100, so that shouldn't be an issue today. But I have been asked to ask you to please not forward the link to anybody because we don't want to exceed the workshop cap. I want to let people know that we are sharing a screen, and so you should be able to see the screen that Brandon is showing. It's a welcome screen. It talks about the things that I just went over about being muted on entry and using the chat box for discussions. And so I want to invite folks to be able to access that either through the app with MS Teams or through your browser. And we did find on day 1 that some folks had more success with the browser. And again, if you want to have some assistance with that, please don't hesitate to email the team at engage@deltacouncil.ca.gov. Okay, so with that, I would like to just, Brandon, if you would advance the slides there a couple, I'll just go over the objective of the workshop again. For those of you who were not here on day 1, and also just to remind folks what we are doing here today, we have convened subject matter experts in order to explore the best possible approaches to accurately estimating the population of spring run Chinook salmon in the Sacramento River drainage. The workshop will help to more accurately inform management actions, including requirements described in California's Department of Water Resources incidental take permit as part of the state water project operations. Next. This is our agenda for today. For folks who weren't here for day 1 and day 2, just to orient you, we did meet in a plenary session in day 1. We had a number of really great presentations on the state of the science, in particular in 3 topic areas: monitoring, run identification, and juvenile production estimates. In day 2, the breakout groups convened on those particular topics and had morning discussions exploring the science available on those topics and gaps, and how they might be incorporated into a JPE. And now we are convening for day 3. We will have presentations of the breakout session discussions so that you'll be able to hear a report out from the facilitators from each of those 6 breakout sessions. They will be just limited to about 5 minutes each, so they won't be able to say, you know, word for word everything that you all put together yesterday morning, but they are going to give a summary. And then we'll have a little plenary discussion on that. We'll take a break, and then we'll come back for Ted Summer and Louise Conrad to give a synthesis of the breakout sessions at a higher level, really drawing it all together. And then we'll again have a plenary discussion on that and then close. So that is our, that's our charge for today. If you could go to the next slide, Brandon. Again, just to remind folks who are just coming in, I saw 25 people just came in. You will start on mute throughout the webinar. And when you want to speak in the discussion, we have a, okay, please use the chat and just indicate your name and affiliation, and then I'll call on you. You can unmute yourself. Again, technical difficulties, contact engage@deltacouncil.ca.gov. Next. Okay, again, I just want to remind folks about the ground rules that we went over on day 1. We're going to have a lot of discussion today, and this is a hard topic, right? This is a very challenging charge that you all have. And so I want to encourage folks to listen and seek to understand each other. Bring your curiosity. Be hard on the problems, not the people. And please try to be succinct, get to the point, be brief so that we can have space for everybody to join the conversation. Next.
Julie, are you there?
Yeah, I'm glad I'm not the only one that lost audio. Julie, we can't hear you.
How about now?
Yep, there we go.
Much better.
I'm getting a little echo. I'm going to
not hearing the echo, so you sound okay here.
Okay, excellent. So then I was just going over the next steps. There will be video available from day 1 and day 3 of the workshop. There will be a summary, including the day 2 breakout groups, and you can find them on deltacouncil.ca.gov/events. The big product will be that there will be a new peer-reviewed scientific article published in the upcoming year. All right. I think I'm going to kick off the presentations from the breakout sessions, starting with Laura Cordoglioni on monitoring.
Can you hear me?
Yes, Laura.
All right. So as a summary, so I would like first to thank you all the participants that called in yesterday for the monitoring breakout session and that actively participated. It was a great discussion. We learned a lot. And I think we have a few points that I'm going to share. And of course, we discussed about way more than that, but just to summarize, the group first think that there is definitely a need for more developed genetic sampling protocol across the monitoring program, and especially because there's a strong overlap of spring and fall run in the watershed. And so using the new genetic tools such as Sherlock or other new genetic technologies that were presented, for instance, on day 1st, would be a preferred approach, as thought by the group, instead of using lanthaday criteria or even untitled models that are developing, because it seems that those models are not properly identifying spring-run juveniles, especially the one that reared in floodplain. So there are challenges with that, such as cost and the ability to trap enough spring-run to run those genetic sampling in a way that it would be powerful enough. But they definitely think that we need to emphasize monitoring and efforts towards that. The second point that I think the group was agreeing on is overall, there is an adequate monitoring of adult spring-run spawners in most of the tributaries, except in some tributaries where there's a strong overlap of spring-run and fall-run adults, such as Feather and Yuba River, and where more genetic identification should be performed there. But so overall, they think more focus should go towards juvenile monitoring, and especially the development of trap efficiency estimates for all the major tributaries or trapping locations that are important for the development of the JPs. And it seems like it's likely feasible, that they were optimistic about it, that the use of tagging, acoustic tagging technology, for instance, will help developing the traffic efficiency, and that the monitoring in the upper watershed or the upper part of the system before juveniles enter the delta is where we should focus our attention, because it seems more challenging to accurately estimate spring-run abundance once they get in the delta, because of, again, challenges that I mentioned earlier, such as more overlap of spring-run with other population of Chinook salmon, other runs, and again, the difficulty to trap enough spring-run there. So focusing on the upper part of the system and making sure we have a good estimation or a more accurate estimation of how many fish are leaving those tributaries and the upper watershed before they enter the delta. They were suggesting that potentially, however, we could also add some other sampling locations that are not existing right now and that could be more important for spring-run. Especially, they were mentioning that there is a lack of monitoring, or the part between the Red Bluff Diversion Dam and Teasdale is kind of a black box, and maybe figuring out if it's possible to trap or to somehow capture juvenile spring-run in this part of the watershed might help having an idea of how many fish get into the delta. Or some trolling in other places more upstream of the trolling location, which is the first one is Sherwood Harbor. So just some ideas about that. Another point that was important is that they overall think that life history strategies need to be included in the JPE, and that we need a better understanding of at what size and time juvenile outmigrates from the tributaries to the delta. Where do they rear? And in which tributary do we observe yearlings? It seems like in some tributaries, this life history might not be important, so we might not need to really focus on that. But in some other tributaries, as what Rachel was mentioning on day first, yearling is a major component of the life history strategy. And so how important are they trying to focus more monitoring on in the understanding the different life history types? And for instance, by using more otolith studies, so collecting otolith and then running some transformative analysis, or through the use of modeling and looking at how life history strategy influence the abundance of the population or its dynamics. So the use of modeling tools was also suggested to be an important part, for instance, to look at the factors that are influencing juvenile trapping and juvenile abundance estimates. So that could be used in coordination with the trapping efficiency studies. It could also be used to look at historical data that were collected in some of the watersheds where monitoring might have stopped now, and that could help to predict juvenile production based on the current escapement numbers that we have for those watersheds. Trying to use this modeling as a inferring tool. Then another point that they made is we need to make sure we really have a good estimation of juvenile survival from the tributaries to the delta. So more tagging studies need to be developed with either wild spring run or Feather River hatchery spring run or some foreign surrogates, so that we have a better understanding and a better idea of how survival evolves from the place they rear to the delta. Where do they have high mortality, low survival? And again, like what are the factors, environmental factors that could influence this survival? And it was suggested that there are some new technologies in terms of acoustic tagging, for instance, that new tags that are getting smaller and smaller that might soon allow fish to be tagged as small as 50 millimeter, so that we need to look at those new technologies and be able to include them so that we have a better survival estimate for this life stage. That was also suggested to use some feed tagging in the tributaries, and that could help looking at movement and survival of juveniles, especially yearling, for instance, in the summer, or some other tagging, such as like maybe coded wire tag for Feather River hatchery. And then it was also thought that it is important to look at fish or include fish from the San Joaquin River into the JPE, and having an idea of also the abundance of juvenile spring run from this watershed arising at the facility, and that will be an important part as well of the JPE. And then finally, as I mentioned earlier, the Feather River hatchery is the only hatchery that produces spring run, and being able to actually use this hatchery for research more than what we've been doing in the past. So using them as surrogate, for instance, for survival study, or in some case when we're not able to collect some data, to just use Feather River hatchery to get this data. So for instance, for estimating egg productivity, if it's we're not able to estimate egg productivity from wild spring run population in the tributaries, we might be able to use Feather River hatchery fish for that. And I think I will end with that.
Great, thank you, Flora. I will now invite Brooke Jacobs to report out on the monitoring breakout session that she led.
Hi, everyone. So we had a really excellent and wide-ranging conversation in my group. I want to thank everyone who participated. I am not going to get a chance to say everything that we talked about, so I just cherry-picked some of what I thought were the biggest topics we discussed. So we talked about current monitoring data that we feel should be included or accounted for as a part of a new monitoring plan that's going to be developed. Just some examples, we talked about existing knowledge and long-term data sets that are available through ongoing work at CDFW. We think we could do a lot better job bringing that forward into the monitoring planning process to make sure we don't reinvent the wheel and we can learn from things that we've already been doing. There was also a prior effort by CDFW mentioned to plan monitoring for adult escapement estimates across the entire distribution of spring run that was interesting, and people thought that should be brought forward and considered. Another example was that monitoring, people thought had been done previously for a long time on the Yuba and Feather rivers, but didn't have access to the data. So that's a data source that could be very helpful to integrate or learn from. And they noted that existing monitoring does occur on these smaller creeks and natal streams, but it's challenged by funding availability, particularly funding across time. So that was a limitation there. The two big gaps that we noted in monitoring that we felt should be addressed are juvenile monitoring and gaps in monitoring escapement and early life stage survival. So I'll talk about both of those briefly. For juvenile monitoring, we noted that it's very difficult to monitor this on unregulated streams where hydrology is flashy. There's a high level of cost because of staff time required and replacing equipment, maintaining equipment. We came up with lots of different ideas to deal with that I don't have time to talk about now, but we talked through some of those challenges. We talked about the need for a better estimate of the proportion of the population that rears in the Yola bypass and how that might change with free rear modifications. We talked about the need to monitor the prevalence and impacts as a result of pathogens and predation. For example, sea shasta is a known problem on the Feather and Sacramento rivers. And there was a discussion of estimating floodplain stranding and impacts on juvenile survival and adult survival as well. We talked a lot about gaps in monitoring escapement and early life stage survival. There's a need for better escapement numbers, better estimates of egg viability, survival, and red superimposition. We had a really good discussion about ongoing work that's being conducted right now as a part of the San Joaquin River Restoration Program addressing these monitoring gaps. And we talked about some of their successes and ongoing challenges. So there's a really good learning opportunity there. Talked about a need to estimate egg to fry survival as they emerge from the gravel, documenting age structure of returning adults. We tend to assume that they're all 3-year-olds, but some portion of them, of the adults, were likely to be 2 or 4-year-olds, and this could be an important data point. We talked about the need for better adult escapement, and we noted that there's lots of different ways that you could do this, and the resource needs vary substantially based on the technique that you use to estimate adult escapement. And so we talked through some of the pros and cons there, which I also don't have time to get into detail on, but they'll all be in the notes. And we talked about ways that we could combine different survey methodologies to balance the trade-offs associated with data quality and resource needs for the different approaches. My group, as Flora also mentioned, talked about the need for better identification of runs. They also talked about the need to better differentiate between fall and spring run because of overlap and spawning timing. This creates difficulties in monitoring. They also talked about the challenge of differentiating between yearling spring run and winter run as being another challenge. So they suggested better integrating genetics with monitoring to confirm run ID and improve the quality of that data. We also talked about overall monitoring needs. There's some interesting conversations here about we need to think about the ability to integrate different kinds of monitoring and methodologies when planning and implementing a JPE monitoring program. We need to have good access to data scientists and plan and collect the data in a way that's easy to integrate across studies. SACPass was provided as an example of an existing platform that does this that could be expanded. We talked about the benefits associated with getting overall abundance estimates by combining data sources instead of maximizing data collection for any one particular life stage. Other limiting factors that were brought up were the need to protect staff safety and fish health during monitoring activities, obtaining take authorization for needed monitoring, and also access to tributaries and streams to conduct the monitoring. A little bit over time, a very fast overview of a fascinating conversation. Thank you.
Thank you, Brooke. I will now invite Pascal Gertler to kick off the summary from her group on run identification, and then we'll move to Brett Harvey's group.
Thanks, Julie. And I'd also like to thank my group for a really great discussion yesterday, Run ID group number 1. We tackled all of our questions in addition to some Mentimeter ones. So in reference to what is the common understanding for run identification tools, we know that there's a trade-off between genetic resolution and rapid results. And we discussed that the scope and objectives are important when choosing the best ID methods. We also discussed that a tributary-specific ID is important for broader conservation goals, but run-level ID might be adequate for the JPE, especially if Feather River hatchery and San Joaquin reintroduction fish can be distinguished from the independent Sacramento River populations, which is an area of active research. We also discussed some must-haves for any proposed ID method. They must consider how much time or effort the ID method adds for processing fish, especially at juvenile trapping facilities where there might be large numbers at one time. There also must be broad support to fully implement, understand the needs, and coordinate across locations, whether those are sampling locations or water operations. And there must be time to properly develop and scale up any method, which would include training, improving protocols, making things more user-friendly and adapting to specific locations and objectives. One potential solution would be to use the probabilistic length at date tool to determine which fish require further genetic analysis and cut down on some of that processing time. So combine length at date with genetics. When we discussed the critical gaps, there was some consensus that we need to sample before delta entry. We also discussed a more minimalistic approach where you would focus on lower river main stem sites, but we decided or discussed that we've learned a lot from places like Knight's Landing that those main stem sites locations can be very challenging to sample, and that there was also some acknowledgement that broad tributary or landscape scale sampling would be cost prohibitive and likely require coordination to modify and strengthen the current salmon sampling network to focus on what is needed for a spring run JPE. We also discussed life history diversity as one of those gaps, and that it's important to consider in the context of the JPE. Because life history diversity is related to habitat availability, which varies across the tributaries, another benefit to sampling before the delta in those tributaries is maybe to better understand some of this life history diversity. Another data gap is spring run non-natal rearing and adult spawning that strays into less typical streams, and that those things need to be resolved in order to model year-to-year variation in population size. Identifying life history diversity may also help better understand migration behavior of juvenile spring run and inform passage. There was also some acknowledgement that tributary ID monitoring would require adaptation through space and time. When we were discussing trade-offs, the identification of San Joaquin reintroduction fish came up several times, and was decided that it's an issue that is unlikely to be resolved with genetics at this time, and that once they begin natural spawning in the San Joaquin, we can no longer rely on CWTs. There are a number of trade-offs associated with this. One of the trade-offs is just focusing too much on San Joaquin fish when Sacramento Basin is our target. Another trade-off is within the hatchery, we discussed the splitting of egg lots. If they're split in the hatchery, then we could use a genetic tool called parental-based tagging to distinguish Feather River hatchery from San Joaquin fish, but that could conflict with goals for maximizing genetic diversity at the hatchery. Some of the potential non-genetic solutions we discussed was making a San Joaquin specific length at date with hatchery surrogates to better understand in-season migration behavior, or a number of different branding and tagging methods that we discussed. Another option could be to workshop some San Joaquin specific triggers that could help the salvage facility anticipate those fish. And that is all I have. Thank you.
Thank you, Pascal. I will now introduce Brett Harvey for Group 2 on run identification. Do we have Brett Harvey from TWR on the line?
There we go. Can you hear me okay?
Yes.
I had to plug in my microphone earlier.
You're okay now. Thank you.
Okay. Start my timer. Hi, everybody. So I just wanted to say, first of all, well, thanks to everybody in our workshop group. But in the interest of time, I'll just say that, interestingly, our group coalesced around a lot of the same conclusions that Pascal's group came to, and I'll just try and hit some of this I didn't have time to talk about that maybe their group talked about within that time.
You know what, Brett, yeah. I'm sorry to interrupt. My audio of you just deteriorated, and I want to make sure you're either close enough to the mic or maybe have a mic or a headset, just because I want everybody to be able to hear. Yes.
How about
okay.
Now?
Thank you. Great.
All right. I switched my microphone. So one thing that that we discussed was that you're going to need some kind of an ID at just about every stage of the monitoring process, no matter what kind of monitoring you end up doing for the JPE, whether it's adult stages or looking at reds, or you're going to have to determine if that run is a spring run. And so And you know, besides genetics and the length of date, there's just simply like for instance, when the adults are coming in the summer, they're temporarily segregated from other fish. They come in, they're a phenotypic spring run, so they're a spring run. So you can pretty much say they're a spring run. In streams where you only have spring run in particular areas of the stream primarily, you can assume that most of the juveniles are spring run, and then if you need to tag them for whatever reason, you can trace those tags further downstream, and then you're a spring run. So there's various ways to ID the fish that aren't just genetic, but somewhere along the way, you're going to have to determine what those fish are. One of the non-genetic methods that folks seem pretty sure about was actually video tracking and video ID of spring run. That it's an effective tool, that it could be refined for our system. It has, you know, different systems are going to have different nuances, but using morphometrics, you could come up with sex and fecundity estimates and things like that simply by taking pictures of the fish, the big fish when they're coming in. Of all the genetic approaches, of course, everyone's really excited about these CRISPR-based methods, the rapid return, and just how cost-effective they could be, resource efficient, customizable, scalable, rapidly scalable up to a larger watershed, given the caveats that Pascal talked about with training. And in particular, useful with the probabilistic length of date. And Pascal touched on that somewhat, that you can use the length of date to reduce to pinpoint which fish you want to actually target for genetic testing. But similarly, the genetic testing will update the length of date probability estimates. So they can work hand in hand. Alternative ID approaches. So I think I touched on a number of this alternative ID approaches that came up in our group. And one thing that was also brought up by Daphne Gill, our geneticist, one of our resident geneticists on the group, was this opportunities for efficiencies. In particular, you can pool samples. So you say you have a group of 50 or 100 juveniles on the shoulders of the migration season, and you can pool those samples and look for a spring run allele in that pool. And if you get a hit, then you can go back and sample individual fish. And you can do that through splitting your samples initially and pooling the first half, and then the other half, reserve the other half for more refined testing. But it would mean a lot fewer genetic tests. And I think someone in the monitoring group was mentioning the winter run. Similarly, in our group, it came up that the winter run and the spring run are going to be mixing more and more as spring run gets established in Battle Creek. And so there's efficiencies there for genetic testing to track both of those runs. Regarding the San Joaquin River, Pascal was talking about the parental-based tagging and the requirements it'll take to segregate the hatchery broodstock that goes to one river versus the other, so you don't have the same parents in both rivers. But one thing that came up in our group is that currently, most of the San Joaquin River fish that are caught as salvage are hatchery fish. There's very few of natural origin, and even with that, they comprise a really small proportion. Oh, that was my timer. A very small proportion of the take at the salvage facility. So that might not be a huge issue right now. Similar with non-natal rearing, you know, maybe we can do some studies and find that that's not non-natal rearing in non-natal tributaries isn't a huge deal, and we don't have to focus a lot of energy on that. Permits that were discussed in conjunction with monitoring, and we were talking about that. Well, is IDing the fish and taking genetic samples or tagging them in some way going to increase your level of take? And the group was pretty, most of the people that with experience with both catching and tagging the fish said, like, the tagging portion is not going to add that much take. It's really, most of the take is going to be happening just capturing the fish in the first place. And then just the last thing I wanted to bring up is there's the heterozygote. So you have the spring run, the fish that are genotype of spring run, you have the fish that genotype is fall run, but then you have these crosses. And when we're doing the genetics, it's not going to tell us what the phenotype of the fish is. It's going to tell us that, okay, this fish has something that both of the parents. How are we going to deal with that? That's something that really needs to be addressed. And I believe that's it. Oh, there's just one other thing that came up, and it's not necessarily an ID issue, but it was just that it would really help to have sort of redundant JPE estimate methods where, and I think that Ted and Louise are probably going to talk about this a little bit more. Maybe the JPE group will, but just this idea that you can actually get a JPE using adult fecundity initially, and then as the season progresses, you can have further and further refined JPE estimates through monitoring different life stages. But I'll leave it at that.
Great, thank you so much. I'm going to start introducing the leaders for the JPE approach breakout groups, starting with Pete Nelson, and then we'll go to Towns Burgess.
Okay, thank you. So I'd like to thank my great breakout group. Can you hear me? Okay?
Yes, we can hear you now.
Okay, good deal. So I'd like to thank my breakout group, and I'd like to thank my my colleague Jada, who was incredibly helpful in terms of taking notes and keeping us on track and organized. My apologies too to the breakout group. I'm not going to be able to present every concept and idea, but I'll try my best to summarize what we discussed and what we came up with. So perhaps most importantly, there was tremendous emphasis on the importance of identifying specific goals and objectives for the JPE. Objectives that should help us limit efforts or define efforts geographically as well as logistically and conceptually. So, for example, a clear framework should help determine which tributaries are included, should account for and San Joaquin fish, and of course also address the appropriate management objectives. There was also broad interest in a JPE that would serve some fairly general objectives, a JPE that would contribute to multiple management, conservation, and scientific goals. We talked, perhaps the bulk of our discussion was focused on what goes into a model and what goes into a model and what doesn't. So again and again, as other folks have mentioned, as other breakout groups have already addressed to some degree, there's a necessity to evaluate the tributaries as well as main stem Sacramento production. Monitor.
Pete, I am not hearing you now. I'm wondering if maybe you want to turn your video off and see if the audio is clear without the video. Brandon, can you hear Pete?
No, I can't hear anything. But yeah, usually if the video's on, the bandwidth slows down. So if the video's turned off, that should hopefully pick up his audio a little better.
Okay. We'll just wait a second here to see if he can come back on with his audio. You know what I'm going to do is, since it sounds like we're having a little bit of audio difficulties with Pete, I'll move on to Towns, and then we'll come back to you, Pete. Towns, can you unmute yourself and pick up with your group 2 on JP, and then we'll come back to Pete.
Sure, absolutely.
Thank you.
And I'd like to start by thanking all the participants in my group. We had a lot of interesting discussion and highlighted many points that I think will go into consideration for the next steps in producing this JPE. A lot of what we discussed touched into other areas that have been discussed so far today. So I'm just going to grab some additional highlights that weren't brought up and maybe emphasize some things that have already been stated, but just to put some emphasis on these points. So regarding the common understanding for putting together a JPE, as many have mentioned so far, we understood that this was to be focused on the Sacramento Basin spring run, and that the San Joaquin Basin would, not be dealt with in this JPE production. But we think there's some challenges that are still associated with the spring run. Several people have brought this up so far in the monitoring and in the genetics point. And I'd just like to highlight that as we use this JPE for managing entrainment or any sort of export control in the Delta, that in the Delta in particular, the San Joaquin Basin fish that could be in there will present a particular challenge that we're going to need to think carefully about in how we actually use this JPE. It was our understanding that the JPE needs to provide an early warning system for managers as they may handle Delta exports. And so in the design of monitoring and considering what monitoring data gets incorporated into the JPE itself, we need to really think carefully about where those locations are, and are they going to provide the adequate time to respond, as that early warning system for the fish that may be entering the Delta. We wanted to highlight that we're not, when it comes to the monitoring, we're not required to select a single method for all the tributaries or in the main stem, that multiple monitoring methods can be used, and there's various ways that these could be combined statistically or incorporated in a model to provide a robust juvenile production estimate. Another thing that we were considering, and that this was brought up multiple times, is that the monitoring that we'll be doing for this can evolve within a season and over years. And we've seen this with the JPE that's been produced for winter run in the past, as they've used different techniques to estimate egg to fry survival or the juvenile production at different locations. And so we should be flexible in this approach and understand that that there is a potential for these techniques to evolve over time. As far as gaps, again, this has been brought up multiple times, and we'll just highlight it again, that we feel like the genetics could potentially be a critical gap. At least in our group, we thought that there was some risk that could be associated with relying too heavily on something like a length at date model, and that genetics in some fashion is going to be a real critical component to this, and that this is something will require that we dig into it further. Another thing that we discussed is some of the differences in timing and behavior of the young of the year fish versus yearlings when it comes to monitoring. Historically, they've been underrepresented in the monitoring data, and in that sense, particularly due to the timing, it's probably going to be recommended that the yearlings and the young of the year are handled separately as far as the production estimate and their timing entering the Delta. The people in our group felt that annual escapement for many of the tribs was there's a good, robust data set existing for that. Of course, there's always opportunities for improvement and expansion to other locations. But and they also feel like there's at least a general understanding of survival. And these are some of the major components that would likely be used in calculating the JPE. What we feel like is we're clearly coming up short on is what the actual production is coming out of these tributaries. We also highlighted that some of the existing monitoring may require modification to be more useful for the JPE. So as we said, these may evolve over time. And as an example, it was brought up that the feather and the Yuba escapement is mixed. It's mixed between fall run and spring run, and so in this particular case, we may need to do something like using the Vaki River Watcher to separate out the escapement for those two runs. And then another thing that we talked about were these the utility of including the dependent streams in the juvenile production estimate. And our group did see value in that. One of the things we were thinking, because in some years these dependent streams have larger populations that may be making a significant contribution. In other years, the adults entering these dependent streams are very low. And so perhaps one thing we would want to do is have some type of monitoring that includes a trigger where then we might want to increase monitoring in those dependent streams instead of putting a lot of effort in there up front. And then we spoke quite a bit about redundant monitoring. There's some concern that if we are dependent on a particular type of monitoring for a component of the JPE, and that technique, for whatever reason, is unsuccessful in a given year. So, what comes to mind are aerial surveys. If there's some issue that prevents us from conducting aerial surveys and we're fully dependent on that method, we could be missing that component. And so we feel, having redundant methods would be important. And then overall trade-offs, we discussed that there's inherent trade-offs to having a generally simplistic versus a detailed approach, or focusing on cost versus accuracy, and that it's important moving forward to outline what those trade-offs would be, and then consider phasing in new components as the data needs justify additional collections. And then overall, for trade-offs, we discussed that some of these new long-term monitoring plans can take a long time to fund and to design and to permit. And again, as I was just saying, our recommendation coming out of this group is to phase in new data over time, but perhaps what we could do is start with some of the more robust data sets. If we feel that the adult escapement and some of that information are some of our longer term, more robust data sets, that would be a good starting point. And then phase in new data as as it's needed and required.
Great, thank you so much, Towns.
You're welcome.
I want to see if we can go back to Pete Nelson to continue on his report out from the breakout group number 1 on JPE approaches.
Julie, can you hear me?
Yes, I can hear you.
Oh, my goodness, something works.
Wonderful. Congratulations. I know everybody has experienced the same, so you are in good company.
Okay, well, I apologize for the hassle. Here we go.
That's okay.
So, I think perhaps above and beyond everything else, there's a
We lost him again.
Oh no.
No.
Okay. Pete, are you there?
Pete's on mute.
Oh, he's on mute. Pete, would you unmute yourself? Okay. It seems like we might need to troubleshoot this on the side, and we will move into the plenary discussion about these breakout group report outs. And when and if we can solve the audio for Pete, we will certainly invite him to report out on what he has. So want to see if we can spend some time. Now I've heard the report outs from the different groups. I'd like to start with the plenary discussion back on the monitoring, and then we'll go through Run ID and JP, just spend a little bit of time on each one. And I'd like to again remind that use the chat on to indicate if you, I will keep and let you know who's up next, and we'll just roll forward that way. You don't have to type out your whole comment in the chat, but feel free, just put your name and affiliation, then I can call on you and you can unmute yourself.
Hey, Julie, can I jump in?
I want to also just offer, oh, yeah, yes, go ahead.
I'd just like to, if you are on the phone listening to the webinar, to please be sure to mute yourselves on your own phone. It's a little harder to know if background noise is coming from phone numbers or not. So just please be sure to keep track of whether you're on mute or not if you're on phone.
Great, thank
Julie,
you for that
I have a
reminder.
comment. I have a comment too. This is Tracy McReynolds. I turned on my chat and it says I can't send a message because I'm not a member of the chat group. Do we all have access to chat?
It sounds like the folks who have called in, I understand, would not have access to chat. So what I would invite you to do, because we have so many people and I have experimented with unmuting everybody and it is not good. I would invite you to email engage@deltacouncil.ca.gov, and I am in touch with the note taker and they can let me know that those of you who are on the phone would like to get in on the queue for the conversation. So we'll do a little workaround there.
Thank you.
I'll say, sure, thank you for bringing it up. I'll just say that email again. It's engage@deltacouncil.ca.gov. Okay. I just got a message that Pete Nelson is back with us. So before we dive into the plenary, let's see if we can get him online. Pete, can you test your audio?
Yes, can you hear me?
Yes, great. Good to have you back.
Okay, well, hopefully third time's the charm, right?
That's right.
Okay, since you've all heard, my primary message, I think 3 times now about having clearly defined goals and objectives, I'll move on. There was also broad interest in a JPE that would contribute to multiple management, conservation, and scientific objectives. In terms of model formulation, there was, of course, a well-recognized need that we evaluate the tributaries and prioritize monitoring efforts and sort of their relative importance to a JPE. But also, it was emphasized that the main stem Sacramento is potentially an important site of spring run production, and that this is a factor that should also be investigated. There was also repeated emphasis on the importance of accounting for both Feather River hatchery fish, as well as fish from the San Joaquin River. It was broadly recognized that life history variability in spring run was a critical factor, in particular how we account for yearlings. The point was made that despite the rarity of yearlings at salvage facilities, there's good evidence that that this life history is important ecologically and evolutionarily. And so particularly in some critical years, we've got to have a way of accounting for yearlings. In addition to monitoring critical monitoring programs in the upper portions of the watershed, the point was made that there's real value in some of the historical data sets, and that there ought to be ways of incorporating some of those historical data, as well as sort of downstream monitoring programs, at the very least in such a way that would complement upstream or the near real-time monitoring in the model. There was recognition that all of these methods come with an array of errors and variability. That it was important to test these models and how they respond to some of those error estimates. That there was a whole host of additional points or discussions that sort of don't fall into sort of these broad headings of management planning or model formulation, but nonetheless were, I think, important. I'll run through a few of them quickly so as hopefully not to get us too far off track time-wise. In particular, it was broadly recognized that we need clarity on the logistics of timing. In other words, when must an annual JPE be available for management? But it was also noted that these timing requirements at least by the ITP, by the incidental take permit, are not set in stone, could potentially be modified. Adult monitoring programs represent a robust source of data. They're available now. This should be factored in. There was also an interesting discussion on sort of, the relative merits of two alternative approaches. Either you monitor each tributary and apply survival estimates specific to each of those, or, perhaps you develop a metric from closer to the delta that integrates all across all the multiple watersheds. So this led to a discussion regarding, sort of the proximate and ultimate objectives of the JPE, including maximizing limited resources, as well as the potential contributions of a JPE to conservation objectives. It was pointed out that there may be value in trying to account for sort of a sprawl run, so to speak, these, introgression between fall and spring run fish. There is interest in the potential application for some of the new genetic tools. Another important point, or interesting point at the least, so-called independent and dependent populations are actually based on historical data, and things have probably changed. So this is an aspect that needs to be considered as well. There was interest in considering loss triggers for multiple systems to protect the most limited populations. There was also a sort of a brief discussion, but interesting discussion on the potential use of tools, things like habitat production models, monitoring ecoregions with multiple spring run populations for greater efficiency, integrating some of the temperature profiles from some of these different tributaries to account for how changing conditions or variable conditions may impact spring run populations. And finally, it was suggested that we might develop a Chinook spring run life table, sort of, to identify sort of key factors that affect mortality at different life stages. Then the critical points, if you know those, if you can identify those, this may help to optimize the monitoring strategy. I'll wrap it up there, and thank you for your patience with my my technology issues, and thanks again to my breakout group.
Great, thank you so much, Pete, and thanks for your determination and getting your audio to work for all of us to hear the report out. I know we've all been there in the recent months, so thank you for following through on that. Okay, so now I think we will dive into the plenary discussion. And please let me know if you'd like to make a comment using your chat function, or go ahead and email engage@deltacouncil.ca.gov. And the folks over there at the webinar team will let me know that you would like to comment. Again, you don't have to put your whole comment in the chat or the email. I'd like to hear your voice. So we're going to start with monitoring. Any additional thoughts now hearing this distillation, this summary of what came out in the breakout groups? Anything, for instance, that you think, oh, gosh, I really want to delve into that a little bit further, or, oh, this is a gap? I'd like to hear from the larger group. I'll let people find the chat function is up in the upper right-hand corner if you are on the webinar. And I'm tracking my text to see if I get any notice. Let's see, I think I have a comment from Barb Byrne with Nymphs. Barb, would you like to unmute yourself and go ahead?
Morning, everybody. I wondered if either of the monitoring groups talked about current and future funding options for all of this.
Thank you, Barb. That's a critical question, I'm sure, for everybody. Let's see if we can pass that over to Brooke Jacobs. Brooke, do you want to speak to that?
Hi, Barb. Thank you. That is a really good question. And we did talk about that actually at several points during our breakout group conversation. We noted that there's a lot of good monitoring that's going on right now that is subject to variation in year-to-year funding availability. So that's an ongoing problem that we would want to talk about when we're planning the monitoring plan. So that was a big one. And then we talked about resource needs, so funding, for staff and equipment, as being one of the key limiting factors when planning the monitoring program. And we thought through some creative solutions to how you could balance data quality with funding needed to implement monitoring, to combine different approaches to still get the same outcome with possibly slightly lower expenses up front. And I did not have time to get into those, but I'm happy to chat at any time. So that's mostly where it came up.
Thank you, Brooke. Let's see, I have a few more folks in the queue now. I have Brad Cavallo with Kramer Fish Sciences, and then Mark Gard with U.S. Fish and Wildlife Service. And so I'll start with Brad, and then we'll go with you, Mark. Brad, would you like to unmute yourself?
Yep. Thanks, Julie. I just want to make an observation about the juvenile monitoring that I don't think was emphasized much in the summaries that I heard, that there's a lot that goes into getting a reliable juvenile production estimate, particularly when we're targeting multiple life stages. We want to not just do fry, but par and smolts and maybe even yearlings. And it's more than just doing, kind of a trap efficiency test. It's actually a very challenging process. It's very doable, but it requires a rigorous sampling design so that you're sampling at representative times. And that includes high flow events, and then having lots of efficiency experiments for all the different sizes of fish that you're interested in under all the conditions that are out there. And I just haven't heard that emphasized a lot. And there's a lot of literature and examples of how that needs to be done, but it's not a trivial matter.
Thanks, Brad. With that in mind, and as you said, there's existing literature about that, would you recommend any particular next steps about synthesizing that literature or making recommendations based on that literature that could inform the JP?
Yeah, I mean, I've got a bunch of stuff I've written up. I think some of it actually came into the planning groups, but.
Okay. When you say planning groups, the breakout groups, it was brought there?
I don't know. It's getting to be a blur now. I actually meant before this workshop convened.
I see. Okay. So some of it has already been provided for the development of the briefs that were put together for this workshop.
Right,
well,
but
thank you for bringing it up again.
Sure.
Okay, so I think we'll move on to Mark Gard. And I said U.S. Fish and Wildlife Service, but I'm getting a note from CDFW. Mark, would you like to unmute yourself?
Yes. Yeah, I actually moved over to CDFW in January from Fish and Wildlife Service. Just following up on the Vacky River Watcher comment, I think that's a great idea, but I think for the Yuba, it needs to be supplemented with getting some genetic samples on carcasses early in the season downstream of Daguerre. Back in the day, we did find a few reds that were spring run timing below Daguerre. So, most of the spring run we did find upstream, but just something to kind of add into that.
Great, thank you, Mark. With that addition, could you just clarify if those are existing surveys you're suggesting, or would they be new?
I don't know if currently if the carcass surveys on the Yuba, if they collect genetic samples or not. So.
Okay, so something to explore and look into.
Exactly, yeah.
Okay, thanks for that clarification. Okay, I had started the conversation off with monitoring. I'd like to move now to run identification. Pascal and Brett had reported out from the breakouts on that breakout group. And I'd like to invite now this larger plenary to provide some input. Again, similarly as you have done for monitoring, are there any gaps hearing the whole summary or anything that you think that should really be highlighted that the group should explore further?
I knew we nailed it.
That's right.
Hey, Julie,
Everything's there. Nothing left to do. I'm sure everybody's typing furiously now that we've said that.
I think Steve Zoig was about to talk.
Okay. Steve, would you like to go ahead?
Yeah, sure. Thanks, Julie. One of the things with the run ID, and I think it came up in our session too, and it goes back to what the goals of the JPE are, is that for purposes of regulation in the Delta, when a fish arrives at entrainment, it's a if it's identified as a spring run, it's going to count against take regardless of what its origin is. So the particular run ID method we're going to use needs to, or what it needs to be accounted for, what it's going to be used for in terms of regulation versus what ecological questions you might want to ask if you're only going to focus on independent populations, or if there's going to be less emphasis on the feather or the Yuba. Like the feather and the Yuba are going to count the same at the facilities, even if they're not emphasized as much in the upstream monitoring. So that should just be considered when determining the resolution of the run ID needed.
Thanks, Steve. I heard in that a differentiation of the goals, right, for regulation versus ecological sort of scientific exploration. Do you have a suggestion of how you would process-wise differentiate those, or sort of reporting-wise where those would go? Is there any more detail you would like to add there?
Well, I think that kind of gets to what the goal of the JPE is. I mean, I could give you what I think the goal of the JPE should be. I mean, it probably depends on your perspective. If you're talking about the, if you're a water manager or you're concerned with entrainment in the delta, then any spring run is important because it's going to count against your take. If you're more worried about understanding the dynamics of the independent populations, then you might not really care about spring run coming out of the Stanislaus or coming out of the main stem Sacramento. So that's what I mean. It goes towards the goals, like what level of resolution you need. If you just want to identify that it's a spring run, then a very simple method could be used. If you want to know basin specific things, then more simple methods might not be able to be used, and you might need to use something like parentage. So I think it all goes back to defining the goals, and that should lead you towards the specific technique you want to use.
Great. And it sounds like some of the groups did talk in depth about the goals. Let's see, Brett or Pascal, would you like to speak any more to that in relation to the breakout groups and how they talked about the goals and the objectives, in particular in the way that it has been framed here in terms of regulation versus ecological inquiry?
Yeah, I think Pascal's group talked a lot more about the importance of monitoring life history diversity. I'll just toss in real quickly that I think initially in this initial planning stage, Steve was, you know, one of the things he was talking about is it's really from a water manager's perspective. I think we're gonna be dealing with the water manager's perspective initially, just trying to regulate pumping around a juvenile production estimate that's strictly based on how many spring run are coming into the delta. But there's long-term goals and objectives and requirements in the interim take permit that where we're supposed to help develop a life cycle model, I believe, and ultimately work toward recovery. So I think we're going to have our eyes on all of those things. But given the really short timeline, right now, I think it's a narrower focus. Pascal, are you there? Did you want to talk a little bit about the life history diversity aspect?
Sure.
Sorry to put you on the spot.
I captured all of that was about life history diversity. We did talk about how the goals are different if you're trying to model extinction risk versus a JPE, or if you're interested in conservation or resilience. And I think that Brett's answer, DWR's answer, is probably best for Steve's question, but we did talk about how diversity is important, and especially in reference to Rachel's presentation, that we live in a very variable climate, and we need to make sure that we're understanding which life history strategies are successful in which years, and that will help us better predict long-term viability.
Thank you both for weighing in on that comment and question. I want to ask Tracy McReynolds and Morgan Kilgore with CDFW, because we did receive emails from you both about just wanting to be able to engage in the conversation. Would either of you like to get into the conversation now?
Sorry.
I don't mean to put you on the spot. I just want to make sure you have access.
No, yeah, I just didn't have access and sent to engage a thing, and I still don't see access. I really didn't know what I wanted to say, but something came up. I just wanted to talk. Brad brought up trap. I think he was talking to trap efficiencies and coming up with a JPE abundance estimates, and how you could do that for key times at key times of a life history cycle to get different estimates, which is great. But overarching all of this is how take fits in, and how your level of effort to get a juvenile production estimate or do trap efficiencies with traps takes a lot of take, and even with genetics. And I guess it's more of a question for maybe Noah at some point. What are the give and takes for take for a species? And I don't know. I think I'd like to hear more about how take is is appropriated for projects, and looking at something that is really heavy on take, like conducting trap efficiencies. What is at the expense of giving a large amount of take for a project like that? And how does that affect maybe other species take against like green sturgeon bycatch for something, a place that's catching both species or multiple species that have take? So I don't know if we have representatives from NOAA on here for take, but effort and sampling across broad landscapes and take jumps out as a problem for me.
Yes, thank you for that, Tracy, and I'm glad we could get your comment in here. Would anybody, I'm thinking maybe, I want to ask first the breakout group facilitators if you addressed this, and maybe if you had some NIMS representatives in your groups. Brett, do you want to weigh in on this?
I was just going to say that we did address it in our group, but we did not have a NIMS representative, or it's someone who is actually, we didn't have a NIMS representative, but I also don't think necessarily that the NIMS people that issue the take permits would be in this group right now, because there's a lot of compartmentalization of roles in any agency. But it's absolutely correct that, a lot of people are concerned, even now with the level of monitoring that's going on now, sampling has to be curtailed at certain points of the year so that they can't actually reach the level of sampling that they feel is necessary to come up with the estimates and reduce the uncertainty in the estimates that we're supposed to, we're striving toward. So I think that they'll definitely, in the development of this research and development plan for the JPE, there's going to be a lot of conversations with NOAA fisheries. And I know just for other projects that they're trying to protect the species, but they're also very interested in not holding back important research. So hopefully, those engagements are successful and coming up with a good balance. Josh has his hand up.
Thanks, Brett. I have a few people in the queue now. It sounds like Barb Byrne is going to weigh in. I saw Pascal, you're on video. And so we'll go to Barb first, and then Pascal, I'll come back to you.
Well, I am from NIMS, but as Brett notes, I'm not necessarily the one that issues those permits. So I just want to second what Brett said. We do need to consider take, right? Many of these populations are of small numbers, and so we are concerned about protecting all the life stages. So, some of the tools that we can use are things like stopping rules so that you don't, you can give an overall permit for a certain sampling, but then in some situations you may have to stop, and that's been mentioned several times, and that can be problematic, and that we have gaps then in our record in the middle of a migration season, for example. But the flip side is that we get a lot of take. So it's a challenging question. I think Brett said it right. It's just NIMS will have to balance the sort of benefits against the potential costs.
Okay, thank you so much for weighing in on that. And I have Pascal, and then I have Josh Israel from Reclamation after her. Go ahead.
Great, thanks. I was going to point out that Barb and Kathy were on the call, so thanks, Barb. I just wanted to mention that we talked a lot about coordination in our group, and I think there's a recognition that spring run exists across a really large landscape, and we'll need to do a lot of coordination. I also wanted to just kind of represent the geneticists and mention that, that there's swabbing and other options, so potentially, I don't know if Melinda wants to weigh in more on this, but the take could be less invasive than a fin clip with some of these newer methods.
I will briefly weigh in. It is true that it is less invasive. It is still considered take, however.
Okay, thank you for that clarification, Melinda. Less invasive, but still take. And I'm moving on to Josh here.
Yeah. I think Tracy brought up a really good point, and I think that there's a really great opportunity here with the approach that people are taking to planning the science around a JPE. that might yield, instead of having a sort of unlimited, unbounded number of years of collecting information and taking and handling fish for monitoring, to develop a JPE model and then through a series of special studies go ahead and measure the necessary terms to build that JPE. So instead of the concerns that Tracy have that are very important of, this becoming research monitoring in perpetuity, where there's a lot of fish being handled, there being a model and then addressing the gaps in that model through special studies to answer those questions, that can be very purposeful, and hopefully it provides a better approach to the take that will need to be authorized for these kinds of things to get the science that's necessary to build a JPE. And that's just my personal take on this, because we struggle a lot with the idea of research monitoring that goes on in perpetuity and becomes required, and then there's too much take for required compliance monitoring, and we struggle with trying to figure out how to work with the other agencies to balance that. And the way that this is structured hopefully will provide an opportunity for that.
Great. Thank you for weighing in on that, Josh, and proposing more specialized targeted studies, it sounds like.
And this is Tracy, and I just want to respond back to Josh, and that's exactly what I'm hoping we get out of this larger group, is that we kind of think outside the box about how to get to what we need to build a JPE and a model to get what we need without doing the conventional trap efficiencies that are big resource killers and very costly. I think building a JPE and a model and then doing targeted research is the way to be thinking. Like in our monitoring, we really wanted to dial down and dig deep into egg viability for a female, egg production per female, egg to fry survival. So key components of the life history that can drive a juvenile production estimate without having a large amount of take associated it with like a trap efficiency. So I just wanted to tell Josh that's exactly what I was thinking. Targeted research to build a model and look at how effective we are that way, because we really have to address the take issue. We don't want to have a broad landscape of take that takes away from research that we need for other key species. So thank you.
Thanks,
you.
Tracy.
Those folks who have been in this conversation, Josh and Barb and Brett, any last comments on that?
Julie, I think Kathy Marcinkiewicz might have a few additional words to offer from NIMS's perspective.
Wonderful. Kathy, would you like to unmute yourself? We're not getting you, Kathy, quite yet.
Okay, do you get me now?
Yes, we got you.
Okay, sorry. Teams every time changes my mic settings. Okay, sorry about that. I'll chime a little bit. Just kind of follow up a little bit on what Barb said. She kind of hit it. It is a challenge to try to find that balance between take for something that is ultimately giving us information that would help us, honestly, build up a better and, more robust take limit, right? If we have a better characterization of what the population is, that helps us hone in on those numbers. So, it is sometimes a challenge, but, our take determinations for any permit, they're not arbitrary. They are done with looking at the context of the population and identifying, what from the viable unwanted population criteria can the population kind of absorb without being put at higher risk. So that's something that, is important in what we consider in issuing those permits. We want to have that be robust and scientifically backed up, but like Barb said, we don't necessarily want to either stand in the way of research and science that's ultimately in the long run going to help provide more information and be beneficial. no easy answer, but wanted to kind of just follow up on that it is something we can work through, and don't intend to be an impediment, but those limits are there for an important reason, for us to kind of help sustain the population.
Great, thank you so much. I want to go back to Morgan Kilgore. Morgan, I'm not meaning to put you on the spot here, but I know that you don't have access to the chat, and you did email. So would you like to offer a comment on this thread or a new idea?
I will email questions as they come.
Okay, great. Thank you so much. Okay. Yes,
All right.
Flora.
It's Flora. Can I weigh in really quick on that as well? Just mentioning that the group was talking about the effort that was done at Red Blood Diversion Dam to look at trap efficiency and how it relates to covariates such as flow, and trying to develop a model that could maybe potentially predict what would trap efficiency be for different environmental conditions. And so maybe kind of using this approach to look at trap efficiency in the tributaries. So maybe that would mean not having to have very fish expensive or strong effort towards estimating trap efficiency in all the tributaries, but maybe looking at some of the tributaries where you have trap efficiency that is already performed, that is already that is working well, and using this information to maybe extrapolate to other tributaries where it's not done and where we don't want to put more pressure on the population. So it's just an idea as well to just kind of learn from what we know again, and then try to use more mathematical tools to get to where we want to go without using too much tech.
Right. Thank you, Flora, for offering an option to the challenge. This has certainly been a rich thread. Anybody else want to weigh in on this? You can let me know on the chat or by email. I'm not using the raise hand function. I know that might be perplexing to some, but I can't see the comments and the hand raise at the same time. So anybody else on the chat? Okay. Let's just spend a few minutes on any comments on the JPE. Pete Nelson and Towns Burgess had reported back on the breakout groups. Anything on the broader approach that people want to address before we take a short break? Okay, this must be break time then. We will take a 15-minute break, and we will return. It'll be a little shorter than 15 minutes. We'll return at 10:40 for the synthesis of the breakout sessions by Ted Summer and Louise Conrad. So I will see you shortly at 10:40. All right, everybody. Welcome back from our break to the Sacramento River Drainage Spring Run Chinook Salmon Workshop on the state of the science for population estimates. And I would like to introduce Ted Summer to provide a synthesis of the breakout groups. Ted, are you on?
I am on.
Great.
Yeah.
Welcome, Ted Summer, lead scientist with DWR, and he's going to kick this off, and then Louise Conrad with Delta Stewardship Council will follow. Go ahead, Ted.
So the first thing I want to say is just wow, we've been very impressed with the depth of information that we've been provided, and Louise and the team and I are still trying to digest all of this. So what we're going to present now is designed as kind of a short, high-level integration of a lot of the ideas you've been hearing about. But I want to note that a lot of these ideas are going to evolve after we get a chance to discuss this further. And also over time, we expect a lot of this is going to change as some of the research we're talking about starts to generate some results. And finally, I hope everyone recognizes we're going to have to evaluate each of the ideas that we've gotten in terms of a whole lot of criteria, including cost, feasibility, etc. So next slide. Brandon, could I get the next slide? Perfect, thanks. So one of the key things that came up today and in the previous groups is what the goal of this overall effort is. And I thought Brett did a good job covering this, but I really want to reiterate this because it is fairly specific. As Brett said, in our state permit, the focus of this juvenile production estimate is entrainment management. So we need to know the number of fish coming into the delta, and then we need to be able to quantify the fish lost at the diversions. So that tells you that we need to be somewhat focused, but that doesn't mean that a lot of the information that we're talking about doesn't have broader applications and interest. And one of the other goals is making sure the information that we're collecting has other value. So for example, being useful for life cycle models and evaluation of other management actions. The other thing I want to note in the goals here is the importance of timely information. We can do really in-depth studies and monitoring, but for entrainment management, we need this information as soon as possible. So again, this is kind of the background context for what one of the main drivers of this effort is going to be. Next slide, please. So we have some general challenges that we've heard about here. Life history diversity is one of the more interesting and unique aspects of Spring Run that we're going to have to deal with in the JPE. We also heard about the idea that there are multiple tributary sources, and the monitoring effort that we have out there varies a lot across those tributaries. There's also the issue of non-natal rearing. The fish don't behave in a nice linear fashion. A lot of them do wander off into these side areas. In addition, we have a need for timely information. Again, entrainment is a lot of the focus of our effort here. We need to have a heads up about what the size of the year class coming to the Delta is and the relative timing and life stage. And finally, as we've heard over and over again, race ID is this huge issue that we need to make sure that we can tackle. Next slide. There we go. Thank you.
Yeah, it looks like there is a little bit of a delay, Ted, on the slides.
Okay, no problem. So given all of this, the primary purpose of the JPE and all of these challenges, what kind of toolbox do we have out there? Next slide, please. So based on your feedback, I'm going to try and cover some of the key things that we're going to have to integrate into the coming JPE work plan and into the eventual implementation of that approach for management. So the first thing that we want to acknowledge is this is a really hard problem. I hope someone doesn't get the idea that, hey, you just put some nets out there, you count the fish, how hard can that be? This is way harder than winter run. It's one of the harder things that I've tried to approach in my career here. The second thing we want to note is that this is really a multi-tool approach, and Brett and others covered this, but we're not going to be able to just simply monitor our way out of this one. We can't just ramp up the monitoring and walk away. We're going to need an integrated approach that uses monitoring, and as you heard from Flora, experimentation to help support things, and modeling to help us leverage some of the information. We also heard loud and clear that we have a need for some redundancy in our approach. Just using one method is probably not going to be sufficient, and there's going to be a need to cross-compare things. Not in perpetuity, but redundancy can help us evaluate what works best and under what conditions. The other thing that's pretty obvious is that we are going to have to make some compromises. We don't have unlimited resources here. And there are also a lot of issues. For example, as we just heard, take is a huge issue. There are certain things that are going to have to be rationalized to make sure we have a workable approach that's going to work for our main project goals. As you just heard from Pete and others, we need to be able to account for these different sources of fish at different points, in particular the San Joaquin River fish and the Feather River hybrids, the sprawl run, in addition to hatchery fish from the Feather River. It doesn't mean we need necessarily a JPE for each of those sources, but we need some way to account for them when we're considering the overall JPE and in our accounting for entrainment at the water projects. One of the more important considerations that we also heard loud and clear was the need for accounting for different life stages. Spring run have this very creative life history. One of the ideas, therefore, is that we may need a separate approach to the JPE for yearlings and young of the year. That's not ideal from just an overall, modeling standpoint or management. We want to be able to integrate life stages into a single model. However, think back again to our primary purpose, entrainment management. Our entrainment season starts in late fall and goes into the end of spring. In a given entrainment year, we're actually dealing with two separate year classes. One is early on yearlings from the previous year class, and the other is the young of the year from the current year class. So we need to be able to account for both of those sources in a given year. We also expect that we're going to have to vary our sampling via location. And we heard that idea echoed by Towns and other folks. There are a lot of potential locations these fish can be. There is likely to be more effort put in the core locations, in the independent sources, but that doesn't mean that we will not try and also account for some of the dependent locations. And then finally, as we heard from several folks, the approach is likely to evolve. And this is first true on a seasonal basis. So I'll kind of go into this in talking about the JPE concept. But as Brett described, we may start with an initial estimate based on initial information that would then progress as we got better information about subsequent life stages. We may also change our approach annually. And so that relates to the previous comment, the varying effort by location. We may have certain triggers. So for example, if adult spawning is detected in some of these peripheral tributaries, that could trigger additional sampling and estimation approaches for those other locations. And then finally, we fully expect this approach to evolve over time. As Flora noted, we're going to be doing research that can be integrated, and Josh also echoed this. We want to be able to roll up that research to give us some predictive ability and not do everything in perpetuity. Next slide. Thanks. So given that, Louise and I are going to cover some of the specific considerations with each of the 3 types of breakout groups that we had. And this is, again, going to be very brief. And a lot of this is going to be somewhat repetitive, but hopefully you will appreciate seeing this integrated in the overall message. So on the JPE approach itself, the modeling, the analysis needed to support the JPE, we want things to integrate with the existing winter run JPE approach so that it's complementary. We also need our approach to be useful for life cycle models. At the same time, as Flora covered in her introductory talk, and we heard time and time again in some of the breakout groups, we have some special challenges here. There's the run ID issue, error estimation, changing additions, just to name a few. So again, this is a pretty tough nut to crack. Next slide. So one of the things we heard from Pete and others is the need to have some sort of guiding conceptual model to help organize our thinking. So one of the things we did in our breakout group was to have at least a starter conceptual model to help understand the direction we might be going. So here is, again, a starting point. And I want to first point you to the left side of the slide, where we have kind of the standard transition between life stages. So adults are producing eggs, though some subset will survive. We will have juveniles in the tributaries, and some portion of those will survive to the delta. So the first point is each of those different boxes or stages give us an opportunity to develop a JPE. So this is akin to the suggestion that Brett passed on, the idea that we may be able to do an early season estimate based on adults, and that is some of the best information that we currently have that will give us that early warning. But that could evolve then to try and work towards estimates for the tributaries before finally getting an estimate of fish going in the delta. So as I noted, we have some pretty good starting information for adults, but Flora also noted there are places where we need to beef up that information. So as far as management in the delta for entrainment, that's going to be one of the most important areas that we need to get right. But honestly, as Flora noted, accounting for juveniles in the tributaries is going to be one of the harder lifts, and we expect that will be where a lot of the effort, the new effort, will go into. And lastly, I do want to acknowledge juveniles entering the Delta is at the bottom of the list. That's despite the fact that that is our ultimate goal, to get that JPE in there. But because of the timing, it's actually somewhat lower in our priority list. So this gives you a sense of the hierarchy of where we might put some of our effort, and how we might integrate different information sources over time. So again, all of those 3 types of sampling could be used at different points to develop a JPE. And as illustrated by the image, we need to be able to account for the different life stages that are coming down. So the young of the year, as well as the yearlings. And then down in the Delta, we need to be able to account for the portion of fish coming in as juveniles, what fish we're seeing at the State Water Project, and so having an ID technique that can account for that. So that's a basic start. Again, it gives you a sense of how we might approach things and set some of our priorities for the coming work plan. And with that, I'm going to turn it over to Louise to talk about some of the specific components that we heard about from the teams.
Thank you, Ted, and hello, everyone. Thank you for being here. And I am going to, actually, next slide, please, Brandon. I am going to follow on Ted here with continuing this, what I'm thinking of as a layered synthesis. We heard from the facilitators of the breakout groups, kind of a distillation of a very far-ranging conversations that they had. And at about a 10,000 foot level, and now we're trying to move to the 30,000 foot level in order to bring home some take-home points. And again, as Ted said, some of this is can feel a little repetitive, but I think this repetition with the complexity of the topic that we're dealing with is helpful. So I am going to, following on that general framework for the JPE, dive into the themes of the fact sheets and the breakout groups, again, with monitoring, run identification, and juvenile production approaches, synthesizing what we have learned so far over the last couple days. So on monitoring, I think we heard there is agreement that quite a bit of monitoring is happening. The challenge moving forward is that we need to align the effort and the, albeit limited, resources that we have to really address the specific needs for a juvenile production estimate. There has been a lot of discussion about the upper tributaries and how we need to get a better handle on productivity in each of these systems, while at the same time I heard several groups acknowledge that we have a black box in our understanding of survival in the main stem of the Sacramento River. So, and when we think about the full life cycle of spring run Chinook salmon, we have difficult choices to make about what part of the life cycle to start devoting more resources. Where do we enhance what we're doing already for monitoring? And I want to note that there were 2 monitoring groups, and we heard from Flora that her group really wanted to emphasize the importance of understanding trap efficiency and really getting upping the game a bit on juvenile monitoring. However, Brad's comment earlier is really important. This is a really difficult thing to do. Trap efficiency is hard. There's flashy hydrology in these tributaries, and they to get really good trap efficiency, you need to look at how your trap is working in different conditions. Very hard thing to do. On the other hand, Brooke's group on monitoring really dove in more on the importance of early life stages. And so we have some more thinking to do on where to devote more resources. Another really, I think, an area where there was a lot of consensus is that we need a better handle on the contribution of yearlings and how diversity in life history strategy changes or influences which life history strategies are contributing to the population. Yearlings so far are not well seen in our monitoring system that we have. So what is their contribution? How does it vary across tributaries? How does it vary across years and conditions? That's an area where we really do need more focus. On monitoring in general, we're clearly not ready to start limiting our toolbox. We need to look at the tools that are available to us. And we heard, I think there was an important comment that using and leveraging multiple tools with respect to snorkeling, juvenile sampling, incorporating genetic tools is going to help us build resilience in our monitoring program moving forward. Next slide, please. So on run identification, I think there is broad consensus. I'm hearing broad consensus that we need a sharper focus, we need more resolution, and we need this at all life stages. We have, again, an expanding toolbox for run identification, particularly with respect to new genetic techniques and tools. So we need to bring all of these to bear, and while at the same time making decisions about how to spend time and money. And I think Pascal her group brought forward an important trade-off between genetic resolution, say at the run ID level versus tributary, versus getting fast results. So those are going to be other decisions. There was also important notes on the need for better resolution on run identification before the fish enter the delta. So, and one of the benefits, if we can get a better handle there, is that we can bringing that back to the life history question. If we have more resolution on run identification higher up in the system, that may help us to identify how different life history strategies are contributing to the population. Finally, we need to be careful and remember that, yes, this is about, we focused a lot of time in the last few days on Sacramento drainage fish, but when it comes to operations in the South Delta, you can't ignore the San Joaquin River and that there is a program for a spring run there. So this is critical to have identification of Sacramento spring run versus San Joaquin Basin spring run in the South Delta. So as the San Joaquin River program includes more and more natural origin fish, there could be, we increasingly need that resolution, and we need to resolve some uncertainties. I think there's some still rich agreement on the value of genetic tools for that question. For example, how parentage-based tagging can help. Next slide, please. So when it comes to the juvenile production estimate development, this is where we are connecting a lot of dots. And I do like this picture of a brain connecting all these different dots, because it really is going to take all of our skill sets with respect to advancing the science, and then also communicating and coordinating around that science in order to build a robust JPE. So we heard very clearly that we need a clear definition on what the goals of the JPE specifically will be. This is going to, those specific goals are going to be important for decision making. We know the tool needs to be relevant to management. Yes, we know that we've heard the need for an early warning system. What exactly does that mean? When is it that we want that JPE number to be available in order to inform management? How then do we leverage and advance our monitoring network in order to inform availability of that number at the time that we need it? It's critical to have an overall framework in order to start focusing our efforts. I think that it's helpful and as Ted said, a starting point to have this conceptual model that he just presented to start developing this framework. From Pete Nelson's group, there was mention of a life table for spring run salmon, and that could be useful for really, again, another tool for honing in where we have key uncertainties. Development of that will take some time. All of this needs to happen while being mindful of limited resources. Staff, equipment, staying safe the whole while we are doing it. There was a valuable discussion this morning about permits and take, and I think that it's critical to think about take as a resource. It's not simply a piece of paper that gives you permission to do something. It's a resource that is limited. And again, cost, money. We only have so much, so making careful decisions. And that careful decision making is critical to advance our and leverage our muscles for coordination across different groups. There's a lot of one thing that's clear with this large workshop that we've had is that there's a lot of interest, and there are existing groups and programs that are already doing a lot of work. Again, how do we leverage all that in order to advance the science appropriately and in a relevant way to the JPE? Next slide, please. So again, circling back into a large, big picture framework, some initial expectations. We know that multiple tools are going to be necessary. Effort is going to the goal is, I heard this in a breakout session yesterday, that one of the intents is to advance the science and to bring that science to management. So on the right here, I just tried to get a really simple conceptual diagram of kind of how this might work, and starting with more research and development in some of the areas that we've noted where we have uncertainties over the coming years. And that will advance science and monitoring, and then that's where our coordination is really going to be necessary to bring together that science to inform a juvenile production estimate. As we heard from Ted just now and from Brett earlier, that there needs to be an iterative process here. We have to evaluate what we're doing. How did the JPE serve us? What else do we need? And then go back to more research and development as needed. Looks a lot like an adaptive management wheel that many of us have seen before, but I think that's an important framework to think about how we implement this process. At this point, I will turn it back to Ted for him to cover next steps as we move into closing the workshop this morning.
Could we get the last slide, Brandon? Perfect, thanks. So this has been incredibly helpful. You've given us a lot to chew on. As Julie noted, one of the key products from this workshop is going to be putting together a manuscript that can be published so that this is all transparent and reviewed and so forth. But more urgently, Department of Water Resources has a requirement to put together a science plan to California Department of Fish and Wildlife for approval by December 2020. I want to emphasize that this is not going to be some sort of detailed monitoring plan that shows all of the locations, the timing, the budget for each and every element. What we've talked about here is a really useful framework, and we need to have the sort of adaptability that Louise mentioned to be able to learn from some of the R&D with monitoring and tool development and adapt the science plan. So we are going to try and put together something that gives us a good starting point and then a process for how we will get to the point of having a JPE approach. So the goal then is starting this January to begin the R&D on a lot of the elements that you have all talked about, so that by 2025, we can have an approved JPE approach to submit to California Department of Fish and Wildlife. That is an aggressive approach. It's something that the timeline was set by the director of Department of Fish and Wildlife and my own director. So we know it's going to be a heavy lift. We also know that there is going to be a need for ongoing evaluation and refinement. So bottom line, this is not the last time we are going to be engaging you all for input. There is going to be an ongoing need for coordination, outreach, working with folks. So I don't know if every single workshop is going to be structured quite like this. Hopefully the remaining ones that we do will not be online, that we'll be able to talk in person. But I want to thank everyone again for the input we've gotten today. And also thank you in advance for the help you are going to give us going into the future. So with that, I think we have some time for questions.
That's right, Ted. We can now open a little discussion about, in particular, the synthesis that you and Louise have just presented. And I think we'll, again, take questions and comments via the chat or by email, and I'll put people in the queue. We'll give folks a chance to get started with their question or comment. Let me ask this. Seeing the high level summary that Ted and Louise have put together here, seeing it all together in potentially a conceptual framework there, or the beginning of one, are we still missing anything? Is there anything that that seeing it as a whole that you go, wow, we need to get this one piece in there or this one element? So just putting that question out there, it looks like I do have a comment from Bruce Herbold. If you'd like to unmute yourself.
Sure. I can't respond to the question you raised. I did want to say that I enjoyed and learned a lot more than I expected to from this. Really encouraged more than I was before. But one of the things I brought into this was a skepticism about trying to keep the San Joaquin spring salmon out of the issue. And what came clear in the workshop, in the team yesterday, was that we can't do that because they're going to be affecting they're going to be showing up in take, and we need to know where they're coming from and how many to expect, even if they're not really part of take because of the way that project is being developed. But it occurs to me that if we embrace them as part of the spring run that are out there, if the San Joaquin, and I do not know that they do, but to the extent that they represent the timing and survival and response to year conditions, annual climate conditions, they're a lot easier to monitor. And so we'll know when they're entering the Delta. We can sample at Vernalis a whole lot better than we can upstream on the Sacramento. And we've had that going on better, I think. We can sample them coming out of those tributary streams because there are fewer tributary streams if they're coming out of the tribs instead of out of the project and down at Friant. But I think embracing that as an indicator of what to expect, or to correlate with what we're seeing out of Sacramento, which is harder to monitor. That seems useful to me. I'm also easily deluded.
Thank you, Bruce, for your comment. And but it sounds like this idea of they're easier to monitor. How could we use them as a reference, as you said, to correlate with the Sacramento? Louise or Ted, would you like to speak to that, or should we? I notice I don't have any further comments, so I'm just giving an opportunity to speak to that potential.
So I'll try and chime in. So first, yeah, absolutely. I think we can learn a lot from the San Joaquin fish. So for example, Flora noted the idea of developing some kind of statistical understanding of trap efficiency. So we could extrapolate across different locations that are less sampled. Maybe the San Joaquin would be a good location for that. At the same time, again, we have limited resources. So although we can learn from those tribs, we may not put a ton of our own resources into that, but maybe use some of the information that others are collecting from those locations.
Great, thank you, Ted. Any other thoughts on that thread or on the question that I posed earlier about looking across? And maybe Brandon, if you would just put up, there was a slide in the beginning of Ted's presentation, I believe. Is anybody seeing any further gaps that you want this team to know about or address before we move on from today?
Hey, Julie, which slide did you want me to go to?
Let's see if you can go through maybe I think it was the first or second slide past that one. I see Barb Byrne from NIMS. Go ahead. Would you like to unmute yourself?
Hi again. I wanted to complicate things a little further if I could. And I wondered if DWR and CDFW, and it didn't come up in our group, but in thinking sort of 5 years out when potentially the JPE would be used in the context of some sort of maybe entrainment loss threshold. I just looked at the ITP and it seems like the current loss thresholds are generally based on combined loss at both the CVP and SWP. Yet a lot of the operational restrictions in the ITP are specific to sort of the state water projects sort of contribution to conditions in the Delta. And so I just wanted to throw out my sense is that, the San Joaquin River fish, they're gonna, I sometimes use the word contaminate, which is terrible, right? They're gonna show up, I think, more often in the CVP, where Sac Basin fish generally are coming from the north. They're going to more often show up at the SWP. So I just think it might be good, again, for this 4 to 5 year out target, to be thinking about whether or not you might want to have some facility-specific elements of loss thresholds or measures, just to kind of I think you'll have both basin fish at both facilities, but I think to different degrees.
So
Thank
Barb,
you, Barb.
that
Go
makes
ahead,
a lot
Ted.
of sense to me. Yeah, I think you're absolutely right. Since we are collecting data on salvage from both facilities, and within 5 years we should have pretty good race ID information, that we should be able to account for losses individually.
Anybody else want to weigh in on that? Go ahead.
This is Brooke Jacobs from CDFW. I would say that the slate is clean right now in terms of how we might envision a new approach to minimizing take of spring run. We have a lot of work to do in between now and the next 5 years. So, suggestions welcome. I also just want to throw out there that we had talked about the potential of doing a more early warning type minimization measure as opposed to relying on salvage thresholds, which are, in my opinion, reactive and a bit late because they don't account for entrainment into the central and south delta unless favorable habitats. So again, no no clear thinking or clear ideas at this point, but just wanted to throw that out there as another possibility that may evolve over the course of the next few years.
If
Thanks,
I could add as well, Brooke brought up the point that the JPE is not our only tool for entrainment management. We are going to use all data sources. So it is going to be one of the key ways that we can set overall limits, but more real-time management is going to be based on multiple data sources. So that idea of predictive models is one of the more important goals, but there'll be other sources as well.
Great. Thank you so much. I have Brad Cavallo with Creamer Fish Sciences in the queue to comment on gaps. And again, feel free to use the chat and email to let us know if you want to indicate that you want to have a comment or question. Brad, would you like to unmute yourself?
Yeah, thank you. I just wanted to add these summary presentations are really good. And one thing that I noticed was, Ted clarified that the focus of this was related to entrainment management. And so related to that, I think a gap, we have a lot of existing information on entrainment of spring run that we really should be looking at carefully and kind of using that to try to anticipate what our entrainment management standards are going to be for that fish. And then there's also the issue of the genetic data. We have data from 2020 on using the OTS 28 markers for what was spring run. And that analysis could be conducted for some of the previous years. I think all those samples are archived. So there's some existing data that could be mined to get better information about genetic spring run loss at the export facilities.
And Brad, I hope you didn't get the impression from our presentation that everything about the JPE was going to be based on only new information going forward. I totally agree that we can learn a lot from the earlier information that's been collected there. As I mentioned, we're going to have to use a multifaceted approach. And so, yes, historical information is going to be critical.
Great.
And just because I'm always trying to help people share their information, Brad, has that information been shared, or can you make sure that the team at the Delta Stewardship Council has the links to what you're referring?
Yeah. Maybe we can have maybe that's a follow-up on this meeting, like who stuff gets directed to, because that hasn't been super clear to me.
Well, let me get that clarified, and I'll come back in a moment after we hear from Rachel Johnson, research fisheries biologist with NOAA Fisheries and UC Davis.
Hey, Julie, I just took the opportunity to make a comment in the chat related to Barb's good hypothesis to Ted about the San Joaquin fish, and just recognizing that the coded wire tag data for those fish could be probed to look at kind of differential entrainment at the CVP versus SWP and the magnitude of that. Just a comment.
Thank you, Rachel. Anybody want to speak to that in particular? Okay, and I am looking for confirmation with the council team on who should receive materials, and it sounds like you should send them to Eva Bush, and I will. Let's see. I think I had her email here.
We'll put it in the chat, Julie.
Thank you. Would you? That'd be great.
Yes.
So everybody can have it. I know there's some folks on the phone also. So I will get you that email and make sure that I speak it aloud. So probably a lot of you received emails from Eva with materials packets, so you might have it already. But I'm sure the team would appreciate receiving the materials that you all know about. Anybody else for comments or questions on the synthesis?
Hi, can you hear me? This is Jada.
Yes, please, Jada.
Hi, thank you all for the wonderful information and wonderful presentations. I was really impressed with the otolith microchemistry, and I just wanted to follow up to Brad's point. Is there any plans to collect some of the otoliths that are already in storage across the various tributaries to look at the different life history strategies that have been used and to inform our methods moving forward? Do we have any funding or efforts in that direction, or could we make that happen if we don't?
I am wondering, Louise, if you want to address that question, or if you know who, it might be Flora on the monitoring side who could speak to that.
I think that I will ask Flora if she would like to comment first.
Okay.
Yes, we currently do not have funding to look at other otoliths than the one we presented and some Butte Creek otoliths that we're looking at right now. I cannot speak for Rachel and her lab who are doing all the heavy work, but I would be really excited to get funding to look at more spring run otoliths and get a better idea of the life history strategy for other populations. But I'm not sure exactly where this funding will come from and if it's available right now.
Thank you, Flora.
Happy to work on that data if you
Sounds like we have some willing project managers.
We can think about something.
Thank you. And you mentioned Rachel's work. Rachel, do you want to just chime in here about otolith studies and potential funding, if there's any other information that you'd like to share on that?
I'm just echoing what Flora mentioned, that we'd be keenly interested in survey crews who do collect otoliths on the escapement surveys. Some rivers have historically archived them. And so that's where we are able to use the archived collections that they have to kind of reveal those life history strategies. We don't have any existing funding or additional spring run projects to date. I'm particularly keen on Battle Creek. I know that they've been collecting spring running fish for the restoration program, and I think that they've got a long time series and archive sets of data as well. But I would be keen to just learn more about if other people have already started to initiate collections. That's a tool that is relatively new, and so people have collected otoliths primarily for aging, but now that we've got kind of these advanced geochemical tools, we can learn a lot more about habitat-specific growth rates and some of the reconstruction work that we presented of Flora's, just that could be used.
Thank you for sharing that, Rachel. Anybody else with comments or questions on the synthesis? Further gaps?
Looks like Corey Phyllis has a question.
Thank you. Corey, would you like to unmute yourself?
Yeah, actually, I just wanted to add to that conversation about the otoliths. And yeah, I think that the focus so far has been on the adults, reconstructing what the adults did as juveniles. And it gives us great insight into what life history diversity is out there, but we get to see what remains after all the selective forces. So it isn't necessarily a good representation of what the cohort did as juveniles. And what's a missing gap, and it has been identified throughout the last couple of days, is that we don't necessarily have the best monitoring to capture the life history diversity as for the full cohort. So just something to keep in mind when you look at the otolith data. That's what the adults did, the ones that survived in river and out in the ocean. And I would caution it against assuming that that is representative of a kind of neutral selective field. There's probably, and I think we suspect there are years where you're only do better than than fry and the par out migrants.
Thank you, Corey. Again, Flora, do you want to speak to that in particular in terms of monitoring, or is there anyone from the facilitator group that would like to speak to that? And then I have Chris Van Holmes up next.
No, I agree with what Corey is saying. We can look at diversity at two different steps at different time, when there are juvenile fish and what they are doing in kind of real time, and then later on when they come back as adults. And so if you look at the adults, you have all the selection that happened from the juvenile stage to when they came back. So having information on what's going on at the juvenile stage directly is important as well, for sure. And that's what we talked about a lot, about increasing monitoring or increasing targeted monitoring towards all the different life history strategies.
Great, thank you, Flora. I do want to just say I have Eva's email here. If you do have materials or you want to send links for additional information, it would be much appreciated. It's eva.bush@deltacouncil.ca.gov. So for those of you who are on the phone, that's the email to send any materials that this group can collect. And now we'll go to Chris Van Holmes. Would you like to unmute yourself?
Yes. So I wanted to make a comment about the genetics at the salvage facilities. At SACPAS, we already make adjustments based on the publicly available genetic reporting. And if there's archived information there, we can make adjustments to the historical data as well. So we would love to see any archived genetic data so we can report adjustments in using our tools.
Great, thank you, Chris. And Chris, could you let me know your affiliation? I just am not seeing it on the webinar list.
I'm with SACPAS, the reporting tool based out of the University of Washington, supported by the Bureau of Reclamation.
Great, nice to have you. Does anybody want to respond to that in particular or add any additional information to what Chris is sharing here? I know I have a whole team of breakout group facilitators. Okay, I do have somebody else in the queue here, Mariah Meek, and if you could also just let us know your affiliation, that'd be great.
Yeah, thanks. This is Mariah. I'm a faculty at Michigan State University. And I just wanted to contribute to the otolith conversation, just let everybody know that Anna Sturrock and I do have a project that's going on right now that is pairing genetics with otolith microchemistry of Yuba River juveniles. So we don't have our results yet, but we should soon have something to contribute to that conversation.
Thank you, Mariah. And could you talk a little bit about the timeline for that?
Oh, that's a good question. So we just received the samples in my lab, and we were just allowed back in my lab. So we will be running the genetics on them in the next month, I would say. And I think Anna is currently doing the otolith right now as well.
Okay, great. And so if you would please email Eva with any further information, that would be great.
Sounds good.
Anybody else on this thread of having some otolith work that is ongoing or just started? Sounds like the team would like to know.
Hey, Julie, I just wanted to mention that we also collected juvenile otoliths in the Yola bypass, and Mariah did the genetic analysis for those. And I will be briefly talking about the results at the next IEP session next week. And we're hoping to have a manuscript maybe at the end of the year.
Great. Thank you for sharing that. Sounds like there's a lot of interest in it. Okay, any further comments? It could be otoliths, it could be the broader synthesis. Okay. It seems like it might be a good time or indication that we're winding down here. And so I would like to thank everybody for your participation. And I'm going to hand it over to Louise Conrad again to close our workshop.
Thank you, Julie. And I won't take a lot more time this morning. I did want to just say a few words in closing. I just wanted mainly to say I really hope that you have learned something at this over the last few days, something about spring run salmon, a better sense for the population, the run, and the science that is actively being done on it, the management needs and the challenges. I hope you also that you have had a chance to connect with others that you may not have otherwise in your day-to-day. And that you have some sense, although there's a big task ahead for the path forward. And then finally, I want to say a really big thank you to the organizers, the steering committee, and to Julie for the effort put into having us all here together this week. This has been an experiment in some ways, in terms of it being our first very large virtual workshop for the science program and the Delta Stewardship Council. It is also just doing the synthesis effort at the very end, trying to bring together an active conversation so that we have some clear take-home points is also something that is challenging, but a worthy experiment to try. So with that in mind, again, as I said on day one, thank you to the steering committee. You came from many different affiliations and took time out of your normal schedule to participate and give guidance on how to construct these past few days. Thank you again to Henry DeBay for leading the steering committee for the Delta Science Program, to Eva Bush as well for excellent facilitation and keeping us on track, and for receiving all these materials after the workshop. I want to say thank you as well to all of the facilitators and note takers for the breakout sessions. Again, this was something that many of you took on in addition to work you already had to do, and it was not an easy job. I want to just acknowledge that there is a wide-ranging conversation even for specific themes, and you took time to be excellent facilitators, and then just afterwards yesterday to synthesize and how to distill that conversation, and with a lot of support from the note takers. So thank you to all of you. Again, the small victories that we have in having a successful workshop and in for actively being able to log on, as Julie mentioned at the beginning, that I also want to acknowledge we are living through very challenging times. It is a pleasure to be here with you and to talk about one of our iconic runs and species of Chinook salmon with you. So thank you to everyone. Thank you to Brandon and to Brittany Young and to the entire external affairs team at the Delta Stewardship Council for excellent presence here and keeping us all with access to engage here for this virtual workshop. So I really appreciate all of your time and your engagement. Thank you. And with that, I think we are closing the workshop. If there are any additional comments, please feel free to send them to me or to Eva Bush, as you have seen in the chat, eva.bush@deltacouncil.ca.gov. And with that, we will close this workshop and look forward to engaging on this on some of the areas that we've already identified moving forward.
Thank you, everybody.